Okay, welcome everybody. We'll get started. I'd like to welcome you to this Storer Endowed lecture by Professor Ian Hickson. I'll start with a couple of words about Tracy and Ruth Storer. Tracy Storer established the zoology department at UC Davis and was with the university until I think 1975. And Ruth Storer was the only female graduating in her medical class in 1913, and she went on to be the first pediatrician in Davis and establish child health clinics across Yolo County. And so we thank them for this wonderful lecture series. Now I have my cheat sheet on Ian. So Professor Ian Hickson, he got his bachelor's degree at the University of Newcastle upon Tyne. And then he did his PhD with Peter Emerson and Keith Powell at Newcastle and Leicester universities. And then he worked extensively with Peter Emerson on the earliest identification and purification of some recombination proteins in E. coli. And he held an endowed lectureship in the Cancer Research Unit at Newcastle until 1989. And then he joined the Imperial Cancer Research Fund, which is now known as Cancer Research UK. And that's at the Weatherall Institute of Molecular Medicine in Headington, Oxford. And he remained a professor of molecular oncology and the deputy director of the Cancer UK unit in Oxford until this year. And he is literally in the process of moving to the University of Copenhagen to take up a new post which has the grand title of Professor of Molecular Aging in the Center for Healthy Aging in the Department of Cellular and Molecular Medicine. So looking over Ian's CV, I saw that his research career started on a low note. His first paper was published in Nature. And in that paper he identified recombination proteins in E. coli. And he recovered from this disappointing start and has gone on to produce 160 research papers and over 60 reviews and book chapters, and has made many seminal contributions to the fields of DNA repair, genome stability, and cancer. And I think for me what impresses me most about Ian's work is, even though he's a biochemist at heart, he has an unusual ability to switch from the pure biochemistry to mammalian cell biology to yeast genetics and make seminal contributions in all areas. And I think that's really a testament to his skills. And in honor of this fantastic body of work, this year Ian became a fellow of both the Academy of Medical Sciences in the UK and the Royal Society. So we thank Ian for coming 6,000 miles to give this talk.
So thank you very much, Neil, for that very generous introduction. I hope I can live up to it, and I'd like to say it's a great honor to be awarded this prestigious sort of lecturer position here. So, as Neil said, I'm still working in two separate places at the moment. I'm working in Oxford and I'm working at the University of Copenhagen. So this is a picture of lovely Oxford, not taken from my lab, I hasten to add. Unfortunately, I look out over an industrial block of a hospital with a large sort of tower, which is not particularly pleasant. But okay, so I'm going to divide my talk sort of generally into three sections. I'll start with a very general introduction for non-specialists. I'm going to be talking in rather simplistic terms, because that's all I understand, I think, about the role of genomic instability and its role in tumorigenesis. And I'll be introducing this disorder called Bloom syndrome, which is a cancer predisposition disorder caused by a defect in a so-called RECQ family helicase. I'll then talk about the role of the Bloom syndrome gene product, which is called BLM, in the resolution of DNA structures that we think arise both during homologous recombination and during DNA replication. And hopefully at the end I'll be able to have time to talk about the putative roles of BLM and another protein called pitch in suppression and or resolution of anaphase bridge DNA. So these are DNA bridges that arise during cell division. So this is my sort of dumb biologist view of how tumorigenesis occurs and what role genomic instability might play in that. So this is my definition of neoplastic transformation. It's a sort of evolutionary process, a somatic evolutionary process driven by an accumulation of genetic and epigenetic changes that in some way confer some selective advantage. So there are lots of mutation and epigenetic change occurring in cells, but some of those confer a selective advantage. So this cell, for example, is going to have a mutation or an epigenetic change, and that mutation confers an advantage, and that allows for clonal expansion of that population of cells. And that selective advantage could be, for example, the ability to grow under circumstances where negative growth signals are being sent to these cells, but these cells now ignore those negative growth signals. And that process can be repeated a second, a third time, and many, many times until you've got a cancer. Now, tumor cells typically show hundreds sometimes of genetic changes, but there are really only a handful of really important changes that have been defined in the literature, so-called driver mutations, that are crucial for neoplastic transformation. So what are some of those changes? Now, in very general terms, some of them are so-called oncogenes, the famous oncogenes. Mutation generally renders those genes constitutively active, and so many times you only need to mutate one allele of the gene. And they drive tumorigenesis by, for example, making cells independent of growth signals that they would normally need. And they're historically viewed as activators of proliferation, but as I'll show later on this slide, that view has been sort of modified somewhat in recent years. Then there are the tumor suppressor genes, and tumor suppressor genes generally render genes inactive, like the retinoblastoma gene, for example, and loss of function permits, for example, unregulated cell cycle progression, as you'd get with retinoblastoma. And oncogenes and tumor suppressor genes are directly driving neoplastic transformation by permitting cell proliferation, or in some cases, abrogating cell death. And they've been called gatekeeper genes, gatekeeper tumorigenesis genes. Genome stability genes that we work on are rather different, and they've been referred to as caretaker genes. Now, it's known that genomic instability is very common in cancer, but nobody really knows whether that's cause or effect. And genome stability genes, or caretaker tumor suppressor genes, are not necessarily directly involved in or rate limiting for tumorigenesis. They're not really driver genes, as most people think. They're not necessarily selective. So if you mutate a genome stability gene, you simply perhaps increase the probability that you mutate these key gatekeeper genes. So in other words, they're just jumbling the genome up a little bit and allowing the driver mutations to take place. Many of these genome stability genes, not surprisingly, are involved in DNA repair or in the signaling response to DNA damage called the DNA damage response, the DDR. But what's been interesting in recent years is the suggestion that genome stability genes might actually be key driver mutations in cancer. And that's because this DNA damage response is thought to provide a barrier to oncogene-induced proliferation through activating the oncogenes induce a DNA damage response. And instead of that inducing cell cycle progression, instead of that inducing cell proliferation, that DNA damage response actually induces cell senescence. It actually causes cells to stop growing. And so you have to, a cancer, if it's going to continue growing when it's got an activated oncogene, has to override the DNA damage response. And so therefore, mutation of genome stability genes could be crucial for tumorigenesis by this model. So some people argue genome stability genes are just helping to get the right number of mutations, and others argue that they're actually crucial for tumorigenesis. The argument against this is that actually when tumors have been directly sequenced, when the full genome sequence has been done on tumors, the actual mutation of DNA damage genes, DNA damage response genes, has tended to be at a relatively low frequency, and so it seems somewhat unlikely that they're absolutely required. But that's been a prevalent model in the last few years as to why genome stability genes are mutated in cancer. But anyway, if you accept that the maintenance of genome integrity is actually important in suppressing cancer, what are the genes? What genes have been identified? Well, to a large extent, human genetics has told us several of the genes that are involved both in the maintenance of genome integrity and in suppressing tumorigenesis, because there's a whole series of human genetic disorders, and I've shown some of them here, where there's a mutation in a gene that causes a breakdown in the maintenance of genome integrity and a high incidence of cancer or premature development of cancer. So there's Fanconi anemia, for example, and I'll be mentioning that a little bit later. There's the famous hereditary breast and ovarian cancer genes. But the one that we've been working on mainly is something called Bloom syndrome. So what is Bloom's? It's a very rare disorder. So we're not working on it because Bloom's is an interesting condition in itself. We're working on it as a model, hopefully, for genome stability and how it affects tumorigenesis. It's associated with extreme short stature, sunlight sensitivity, fertility problems. They get type 2 diabetes in childhood, which is the late onset type of diabetes generally, associated with obesity, for example, and these individuals are anything but obese. They're usually extremely thin. They have skin pigmentation abnormalities, occasional features of premature aging. For example, females with Bloom syndrome frequently enter the menopause in their 20s. And they're predisposed to all types of cancer. It's one of the only disorders, as far as I'm aware, where you see the full spectrum of cancers. You see leukemias, lymphomas, and the common solid epithelial cancers. But instead of seeing them in old age, which is more generally seen in the normal population, you see them early in life. The mean age at cancer diagnosis in Bloom syndrome is only 24 years of age. And many young individuals with Bloom's have multiple independent tumors. Sometimes 4 or even 5 independent tumors. So they're remarkably cancer-prone and the full spectrum of cancers that are seen. Now, the Bloom's gene product belongs to this family, which has become very fashionable in recent years, the RECQ helicase family. So here's BLM at the top in my BLM sort of centric view of life, named after a coli protein that is well known to some of you here, E. coli RECQ. So this is one of the most highly conserved families in evolution. There's a single family member in budding yeast, SGS1, that we work on, as do people here, and then 5 family members in humans. They all have a helicase domain, and for anybody who's a helicaseologist, this is a superfamily 2 helicase domain. Then they have 2 other DNA binding domains that are present in the sort of canonical domain structure, an RQC domain, that's involved in binding double-stranded DNA and has a zinc binding domain and a winged helix domain and another DNA binding domain. And that's present in the yeast protein. It's actually present in BLM as well. BLM looks like SGS1, you might say, in terms of domain structure. But the other 4 human RECQ helicases have either lost domains or they've actually added additional things. So, for example, WRN has added an exonuclease domain. RECQ4 has added this domain that looks like a yeast protein called SLD2, which is involved in DNA replication. But the real interest, I guess, in this family has derived from the fact that 3 of the family members, the top 3 here, are involved in human disease when they're mutated. So I've told you about Bloom syndrome. I'll just briefly tell you about the other 2. Mutations in WRN cause Werner's syndrome. And Werner's syndrome is very different than Bloom's. You don't have any early onset abnormalities. The individuals are normal until about puberty. Then they show progressive development of aging phenotypes. It's probably the best genetic model for premature aging. So this is a Japanese girl aged 15 years of age, where she looks perfectly normal. And this is the same individual who you might think is about 95 here, but in fact is only 47 years of age. So she's got graying, thinning of the hair, bilateral cataracts, loss of skin elasticity, and many, many other abnormalities associated, you might say, with the normal aging process. This is also a cancer-prone disorder, but they get a very limited spectrum of cancers, primarily sarcomas, a few other cancers, but largely sarcomas, which are cancers of mesenchymal origin. Remarkably, the other RECQ helicase disorder is disorders, really, because in fact RECQ4 mutations cause 3 independent disorders. This little boy here's got Rothman-Thompson syndrome, and that causes a whole series of bone abnormalities. So he has no radius in the forearm, has curved forearms. They often don't have any thumbs, which is actually a feature of this Fanconi anemia disorder as well. They get loss of hair, they get loss of eyebrows and eyelashes. They have skin abnormalities. So Rothman-Thompson individuals get something called poikiloderma, which is a sort of lattice structure on the skin, which is a strange rash-like structure. And they're also cancer-prone, but in even more limited range of cancers, they only get bone sarcomas, do Rothman-Thompson individuals. And Rapidolino and Barlow-Gerald syndrome, the other two disorders, are characterized also by bone defects. So RECQ4 really causes bone problems, bone developmental problems, and bone cancer problems. So Rapidolino, again, this is an acronym, so the RA stands for radial aplasia. They usually have either a small radius or no radius at all, often don't have a kneecap, patellar aplasia. Barlow-Gerald syndrome have problems with the fusion of the cranial sutures, and so they have a very abnormally shaped head. But why RECQ4 deficiency causes bone abnormalities isn't known. So we've got really 5 RECQ helicase disorders which have rather different phenotypes. They have cancer predisposition in common, some features of premature aging, particularly obvious in Werner's syndrome. So I mentioned to you that BLM sort of in a domain structure kind of looks like the orthologs in lower organisms. And so you could say naively that maybe BLM does the role of these guys in lower organisms. Now that's very flimsy evidence, obviously. But we actually believe that BLM probably does do the role that most of the roles in human cells that are done by the RECQ helicases in lower organisms. And the reason for that is that BLM, uniquely amongst the human RECQ helicases, has the same binding partners as, for example, SGS has in budding yeast. So the partners are RPA, which is the main single-stranded binding protein, a type 1 topoisomerase called topoisomerase 3, or TOP3 in yeast, and OB fold-containing proteins in yeast, just RMI1, and in mammalian cells, RMI1 and RMI2. So a topo, OB fold, and a multi-OB fold protein in RPA, a single-stranded binding protein. And as I say, this is the only human RECQ that seems to bind this complex of proteins. Now, the RMI proteins are actually quite interesting as they're multi-OB fold, and they look a little bit like a specialized RPA in some ways. So RPA has multiple OB folds. RMI1 and RMI2 have RPA-like OB folds that instead of binding single-stranded DNA, actually are protein-protein interaction domains. So RMI2 uses its OB fold to bind RMI1's C-terminal OB fold. And this OB fold in the N-terminus, which is the only one that's present in yeast, looks rather more like the OB fold in a protein called RECG. So I'll get back to this idea that maybe RMI1 and RMI2 are some kind of specialized RPA potentially. Now, the genomic instability in Bloom's cells is very extensive, but the hallmark feature that's actually still used as a molecular diagnosis is that they have elevated levels of cystochromatid exchanges, which you measure using bromodeoxyuridine incorporation into DNA. So instead of seeing the typical pattern in normal cells where one of the cystochromatids is stained dark and one is light, you get these exchanges between the cystochromatids in these metaphase chromosomes, giving this harlequin effect. And that's very characteristic of Bloom syndrome cells. So a lot of our early work on Bloom's was thinking, how could the Bloom syndrome protein suppress this cystochromatid exchange? What's the specific role of Bloom's in suppressing cystochromatid exchanges? So the first thing one has to address is, where do cystochromatid exchanges come from? So generally speaking, I think it's fair to say that most cystochromatid exchanges arise during DNA repair, in a way. So they're the byproduct of a DNA repair reaction, and they're a byproduct of a homologous recombination reaction, essentially. So here's a very cut-down homologous recombination reaction where I'm starting with a broken piece of DNA. Then you get strand invasion to form this D-loop structure, and in some models you get the maturation of that structure into this double holiday junction, as it's called, these crossover structures being holiday junctions. And it's thought that the holiday junction containing molecules are the source of cystochromatid exchanges. And the reason for that is that holiday junctions are susceptible to cleavage by enzymes called resolvases, or holiday junction cleavage enzymes. And depending on how you cut these, and you don't need to go into the details of this, but depending on how you cut these holiday junction molecules, you can get two outcomes, crossover and non-crossover recombination. And for the purposes of this, you can just imagine the crossover recombination is scored as a sister chromatid exchange, very likely. And what's happened here, instead of this being red and red and black, as you see in the non-crossover, this is now black and red and red and black. You've basically exchanged all the flanking DNA, and in the context of a chromosome, you've essentially exchanged the flanking arm of the DNA. So now you have a sister chromatid exchange. So how might you suppress sister chromatid exchanges? Because that's what BLM does. There's many ways in which you could suppress chromosome SCEs like this. You could actually stop the process happening at all. You could be a completely anti-recombinase. You could be a suppressor of all recombination. And there are various ways in which you could do that. And one way you could do it that has been shown, at least in vitro, is to eliminate these strand invasion events catalyzed by RAD51. You could basically chuck the D-loop structure out, or the invading strand out, and of course you'll never get any sister chromatid exchanges under those circumstances. But what we worked on for many years, several years ago, is an alternative mechanism whereby this holiday junction containing structure is actually processed in such a way that there's no crossover recombination and you eliminate sister chromatid exchanges. And this was worked out by Leonard Wu when he was in my lab. And what Len showed and reconstituted in vitro was a rather bizarre mechanism for how you might process this double holiday junction containing structure. So what Len showed was that what BLM is doing is acting upon these holiday junction containing molecules and migrating them in such a way that they come together. And that reaction seems to require topo3 as well. And if they come together completely and collapse down on each other, they produce this structure where you just have a single-stranded intertwining called a hemicatenane. And the hemicatenane is actually the perfect substrate for topo3. So topo3 is essentially a single-stranded decatenase. So that means it just passes single strands through each other. And this is a very good substrate for topo3. You separate these two DNA molecules like this, and in fact, you've never produced a crossover structure, and therefore you've completely suppressed the SCE. So a few bits of information about this reaction, which, to distinguish it from holiday junction resolution, we call it holiday junction dissolution. For reasons that we still don't understand, BLM cannot be replaced by any other enzyme, including human RET-Qs that we found, even though many enzymes can branch migrate these holiday junction structures. There's something special about BLM. Now, you might imagine that the special thing is that it binds to topo3, but we made a mutant of BLM that can't bind topo3, and it still catalyzes dissolution. So, in fact, the two enzymes do not require to be bolted together, at least in vitro. This RMI1 protein isn't essential, but it very strongly stimulates the decatenation reaction by topo3. In our hands, at least, RMI2, this second OB-fold protein, has no major effect on the reaction. RPA doesn't have a very big effect on the reaction. It gives a mild stimulation, at least in our hands. And work done by some famous people in this university, Petr et al., showed that it's conserved with the yeast proteins as well. In fact, it's far more efficient, in fact, with the yeast proteins than it is with the human proteins. So, in fact, this reaction, we would say, is probably conserved in evolution as a way of suppressing crossover recombination. So we're interested in testing aspects of the model, and this is where we've switched to yeast. So we've been using yeast SGS mutants to analyze the role of RET-Q helicases in homologous recombination repair and ask some rather simple questions. Do they accumulate homologous recombination intermediates following DNA damage during the process of DNA replication when you think SCEs would be produced? They are induced really during replication. So we've tried to use yeast to address some of the issues related to whether this dissolution model might be happening, at least in a real organism. It's very difficult to do it in human cells. So one of the reasons we use yeast is you can actually interrogate specific regions of the genome and ask whether structures appear at that particular region of the genome. And we work in a region of the genome close to an early firing replication origin, ARS305. And obviously when cells go into S phase, that origin fires, and then you get bidirectional replication. You can cut that DNA and you can analyze it on a two-dimensional gel. And on a two-dimensional DNA gel, you can see all the different structures that can possibly form, you might say, during replication. You can see bubble structures on this bubble arc, Y-shaped structures on this Y arc, and X-shaped molecules, some of which would potentially contain holiday junctions, on this so-called X-spike structure here. So what happens with an SGS mutant? So if you look immediately after release from a G1 or S into S phase in the presence of a DNA damaging agent, which you need a DNA damaging agent, we use MMS. In wild-type cells and SGS mutants, you see all the replication structures the same. There's no abnormality in origin firing in SGS cells. There's nothing abnormal that we can see early in S phase. If you leave the cells for 3 hours, basically all of these structures disappear from wild-type cells because replication disappears out of the fragment that we're interrogating. You can see replication structures further along the genome from ARS305, but where we're looking, they've disappeared. But in SGS mutants, you get the persistence only of the X structure. So you've lost the Y arcs, you've lost the bubble structures, but the X structure persists. And that X structure is Rad51 dependent. If you take a Rad51 SGS double mutant, you see no persistence of these structures. So SGS mutants later in S phase accumulate apparently a homologous recombination structure. So is there any evidence that this X-shaped recombination structure has got anything to do with the dissolution model? So we've asked the question, do the X structures represent holiday junctions or do they contain holiday junction-like molecules? And is SGS1 actively involved in their removal, as you'd predict from the dissolution model? SGS would be removing them from the genome. So what are the X structures? An outline of the strategy. This is a perhaps rather simplistic strategy, but we've tried to ectopically express nucleases with known substrate specificities in the cells. And of course, we've focused on this class of enzymes that are known to cut holiday junctions, holiday junction resolvases. We compare native with nuclease-dead variants of these enzymes. And we've analyzed two structurally unrelated resolvases, a bacterial resolvase called Rosé, and the more recently identified human resolvase called Gen1. Now, Rosé is the bacteriophage-derived holiday junction resolvase, reasonably highly specific for holiday junctions, holiday junction-like molecules. Gen1 is the human enzyme, has rather modest specificity. It will cut holiday junctions. It cuts other structures as well. The interesting thing is, though, that expression of either of these molecules can, to some extent, rescue phenotypes in a RECQ helicase mutant. And in this case, it's an RQH1 mutant of Schizosaccharomyces pombe. That's the SGS ortholog in fission yeast. So these things can do positive, beneficial things in cells lacking RECQ helicases, which is circumstantial evidence that they're helping to resolve some structure that accumulates in those cells, perhaps. So what happens when you put these resolvases into SGS mutants? So here's an SGS mutant accumulating this X-spike structure following MMS treatment. In this case, we've exposed cells to MMS for 2 hours, and then we're actually allowing them to recover in drug-free medium. So it's 6 hours after they started S phase, and even though they haven't seen any drug, any DNA damage for 4 hours, they still have a very persistent X-spike. If you express Rosé in those cells, the X-spike intensity diminishes dramatically. That's dependent on the nuclease. This is the nuclease-dead Rosé. You've got a good, strong X-spike. And again, Gen1 causes it to diminish. And I haven't shown here the Gen1 nuclease-dead has no effect. So on the face of it, these X-shaped structures that accumulate in SGS mutants are being removed in some way by Rosé and Gen1. The question then is SGS actively removing these structures? And this is where we started to analyze a temperature-sensitive mutant of SGS1. SGS-136. So here I'm showing, you might say, the controls. Here's a wild-type cell. It doesn't matter if it's at 35 or whatever temperature it's at. It doesn't accumulate X-spike structures. SGS deletion cells, we know, accumulate X structures. Here we are. SGS-136, the temperature-sensitive mutant, accumulates X spikes at the restrictive temperature 35, but not at the permissive temperature of 25. So now what we can do is we can let the X structures form at the restrictive temperature and then ask what happens if you let SGS be reactivated, you might say. So we split these cultures into two. Here's the SGS deletion strain. The MMS, the damage, has been washed off, and we've left them for another 4 hours. SGS deletion strain, doesn't matter what temperature you have, the X structure just sticks around for hours, doesn't go. Here's SGS-136. It's been at 35 degrees and accumulated X spikes. Leave it at 35, the X spike sticks around. Lower the temperature to 25 to reactivate, you might say, SGS, and the X spike disappears again. Suggesting, I'm only saying suggesting, that reactivation of SGS is causing these X spikes to be eliminated. We've allowed them to form, and now they disappear again. Now, it could be dynamic processing of these things, but we think, since we've washed the drug off as well, it's very likely, actually, that SGS is actively eliminating these molecules. That's the simplest interpretation, we think. So we then asked us a completely different question, is what makes the X structures disappear in SGS mutants? Now, I've said that they stick around for 4 hours at the drug concentration that we use, but actually, eventually they disappear if you leave them 8, 9 hours. And if you lower the MMS concentration so there's less damage, they disappear rather more quickly. So what makes those structures disappear eventually? So we've asked the question, what resolves the X structure, you might say, in the absence of SGS1? And the most likely candidates are enzymes that will act upon holiday junction X-shaped structures. So if one accepts that the structures are X-shaped, they're sensitive to Rosé and Gen 1, they're very likely to contain holiday junctions of one form or another. And several putative holiday junction resolvases or enzymes that can hit holiday junctions have been found in yeast, and the best candidates are MUS81 MMS4, SLX1 and 4, and YEN1, which is the yeast ortholog of this Gen 1 enzyme that I talked about. So in this case, we've used a lower dose of MMS so the X structures disappear a bit earlier, and we've used, in fact, in this case, I shouldn't switch like this, but unfortunately we've used a temperature-sensitive RMI1 mutant for this, but it works with an SGS1 mutant as well. And this experiment's done at the restrictive temperature, so RMI1 is inactive, essentially. And so in all of the cases we've looked at, in the presence of MMS, we have this enormous X spike, and that's because all of these strains have an RMI1 temperature-sensitive mutant. But in some cases, they're mutated in the resolvase type molecule as well. MUS81 here, MMS4, SLX1, SLX4. And so when you allow the cells to recover in the presence of MMS, recover in the absence of MMS, this time, because we're using a low dose of MMS, the X structure disappears in the RMI1 mutant. It disappears whether SLX1 and 4 are active or not. We can't see any effect of those. But in fact, the X structure doesn't disappear if you delete MUS81 or MMS4, suggesting that MUS81 MMS4 is perhaps the dominant enzyme that removes these structures when SGS, top 3, or RMI1 are defective. And in our hands, we can't find a role for YEN1. We see no effect of YEN1 in this system. Interestingly, though, if you overexpress YEN1, you can actually suppress defects in MUS81 and MMS4 mutants. So the suggestion would be, perhaps, that if we overexpress YEN1, we might even get the X structures to disappear in this double mutant system. We're looking at that. So in our view, these X structures are holiday junction containing or some variant of a holiday junction. And because, what I should have said on the previous slide, the single holiday junction resolvase mutants don't themselves produce X spikes on gels. It's only really the SGS complex that produces X spikes on gels. So although I'm completely biased, we place SGS, RMI1, and top 3 at the top of the hierarchy in terms of removing these structures. if one of these components is defective, MUS81 MMS4 can deal with it, and perhaps down at the bottom is poor old N1 that isn't very good at doing it, but if it tries hard enough, it can remove some of these structures, and if you overexpress it, maybe you can suppress some of those things. Perhaps the same is true in mammalian cells. Perhaps the BLM complex is the main thing for removing many of these recombination structures. MUS81, we think, is involved. Steve West has some evidence to suggest that MUS81 is important in cleaning up some of the things that BLM doesn't work with. And again, Gen 1 is playing a more minor role or a more specialized role or a role at a certain time of the cell cycle. And there's a major caveat here, and that is if there's any credence to this sort of model, you might say, it may only be relevant to structures that arise during repair of replication damage caused by agents like MMS. We're not talking about double-strand break repair here. We're talking about replicating on a damaged template. And probably we're talking about the repair of single-stranded DNA gaps that occur after the replication fork has gone. So these are post-replicative gap-filling structures, we think. So the next story I'm going to describe was started by this guy, a very talented graduate student, Chris Chan. And Chris asked the question, what would be the cellular consequences of a failure to carry out dissolution? And so he thought that the most obvious thing, if you're having repair during S phase between sister chromatids, if you can't carry out this process and you don't clean it up before you go into mitosis, then you're going to have some major problems in separating your sister chromatids when you go into mitosis. So that made him look in mitosis at chromosome segregation problems or sister chromatid segregation problems. And so he started out with a fairly basic analysis of just quantifying the formation of obvious abnormalities in chromosome segregation like anaphase bridging and lagging chromosomes. And sure enough, he actually found that in Bloom syndrome cells, shown in yellow here, you get a very high level of chromosome missegregation, and that can be suppressed to some extent by putting the Bloom syndrome cDNA back into the cells. So Bloom's is clearly suppressing problems of chromosome segregation. But what became much more interesting was when he started to look at where the Bloom's protein was in mitosis. And to his surprise, when he saw these sort of chromosomal bridges and he stained for the BLM protein, so this is immunofluorescence, he started to see BLM actually sitting on the bridge DNA. And it was through sort of quantifying these bridges that he came up with a much more interesting observation, at least in my opinion. And that is that he started to see cells that looked like that down the microscope when stained with DAPI, which everybody would score as a perfectly normal anaphase. Here are the two sister masses of DNA separating, but it's only when you stain for BLM, or in fact for this protein pitch that I'll mention at the end of the talk, you realize there's an enormously long DNA bridge in that cell. This is a late anaphase cell. It's got a giant bridge that has BLM and pitch protein on it. And we call these ultrafine DNA bridges because they don't stain with DAPI or any of the commonly used dyes. Now, these UFB don't stain with dyes, and we don't think they contain histones either. We can't actually find any histone on them. All the bridges contain not only BLM, but the BLM partners, not only TOP3 and RMI1, but FANC-M that binds to RMI1, and a protein called RIF1 that is in the Bloom's complex. This is also called BLAP250. And interestingly, pitch depletion from cells causes loss of BLM from the bridges. So in fact, we think pitch is very important for BLM to be on the bridges. If you deplete BLM, pitch is still on the bridges, so pitch doesn't require BLM. What's very interesting about these, I'm showing this example of this enormous bridge because it's sort of dramatic looking. But actually, early in anaphase, many cells have multiple bridges. I mean, they're present, if you look hard enough, early enough, there's probably bridges in every single early anaphase. So chromosome segregation is nothing like as efficient as people have imagined. In fact, bridges are present, sometimes many, many, many bridges are present in early anaphase. And as anaphase progresses, the number of bridges decreases till occasionally in very late anaphase, you have an enormously long bridge. So we think bridges are actually being removed as anaphase progresses. Now, obviously, they could be breaking. The DNA could just be breaking. or in fact they could be being resolved. And we'd like to think they're being resolved, and we'd like to think obviously BLM plays a role in that, because in Bloom syndrome cells the number of the bridges is actually increased, and they actually persist for longer in anaphase, suggesting there's a sort of active removal of those bridges in anaphase. Now, just to make it more complicated, there are two classes of ultrafine bridge, and they're defined by where they are located in the genome. So here's my picture of a metaphase chromosome, and cells go into anaphase, and you see BLM on anaphase bridges. Now, most of the anaphase bridges, for reasons that we don't understand, actually form at centromeres. So centromeric DNA is an enormous hotspot for anaphase bridge structures, and as I say, for reasons that we don't understand. We do also see chromosome bridges that arise from the arms of the chromosomes. And virtually all of those, as far as we can tell, have a little focus, one on each sister chromatid, for a protein called FANCD2. And this is one of the Fanconi anemia proteins that I mentioned at the beginning. And FANCD2 has its partner protein on there called FANCI. So you see a bridge with a focus stuck on the end of the bridge. And the reason that the focus is on the end of the bridge is the focus is actually defining a locus in the actual chromosome. And despite our route to the identification of these bridges, we actually have no evidence whatsoever that they've got anything to do with homologous recombination. We actually think these are replication-derived structures. We don't think they're recombination-derived structures. And these FANCD2-associated bridges that are on the arms are induced by afidicolin and other DNA replication DNA replication inhibitors. And we think they're unresolved replication structures. And here's a very good example of an enormously long anaphase bridge. Here's the masses of DNA, BLM, or in this case pitch on the DNA, and a little FANCD2 focus on the end of the bridge. So where are these loci that these FANCD2 foci are? Well, they're virtually all at specific loci in the genome called fragile sites. And fragile sites are loci that complete replication very late in S phase or G2, or maybe they don't complete replication at all by the time cells go into mitosis. And they frequently show a break in a metaphase chromosome. And interestingly, they're hotspots for chromosomal rearrangements, including some of those that are found in cancers. And for, again, reasons that we don't understand, one of these fragile sites, FRA16D, is a real hotspot for these anaphase bridge structures. About 1 in 5 of the anaphase bridge structures that have FANCD2 foci on the end arise from a single locus in the genome, FRA16D. So what's our model for how BLM and TOPO3 and RMI1 might be dealing with something that's arising during replication? Well, our model really derives from thinking about what a fragile site is. A fragile site is a region that replicates late or completes replication very late. And occasionally, the cell will go into mitosis and it won't have completed DNA replication at these loci. So here's a little bit of DNA that hasn't been fully replicated. Obviously, you can't pull the sister chromatids apart under those circumstances because it's still, the parental DNA is still intertwined. We would argue that BLM and TOPO3 and its partners disentangle this kind of structure through unwinding followed by decatenation by TOPO3 to allow faithful segregation of the sister chromatids. And we've done two things to kind of get a handle on this model. One is we've addressed an issue of what are the consequences in the next cell cycle for having a failure to sort of remove this kind of structure, and some biochemical testing of the model. So this is a collaboration that we've been doing with Yuri and Claudia Lucas in Copenhagen, coincidentally in Copenhagen, even though I've moved there. And they've been looking at the consequences of these bridges. So we thought that perhaps if the bridges break in anaphase, that the daughter cells would inherit a problem. So here we have an unreplicated piece of DNA, gives rise to a bridge, and that then perhaps gives you a gap or a break in the chromosome, and that might then lead to a DNA damage response. The cell would recognize that there's a DNA damage response. And so what Yuri and Claudia were looking at were so-called 53BP1 foci. So 53BP1 is a DNA damage response protein, and they express GFP 53BP1 in cells. And they made an observation that they didn't understand and then we got together and collaborated on this. And their observation was that newly born G1 cells suddenly have 53 BP1 foci in them, very frequently. So G2 cells often don't. M-phase cells hardly ever seem to see these foci. Immediately, they seem to be reborn as G1 cells. You get DNA damage foci. What's much more interesting in some ways is that those DNA damage foci seem to be symmetrical, as you might imagine from a problem that has arisen from the sister chromatids or bridge-like structures. So if this daughter cell has 3 53 BP1 foci, this cell seems to have 3 53 BP1 foci, etc. And that these foci are induced by afidicolin and treatments that induce fragile sites and induce the bridges. And you can get many, many 53 BP1 foci, and they sit through the whole of the next G1 phase till the cell goes into S phase, and then they seem to disappear. And recently, Yuri's got some evidence that these are somehow protecting the DNA damage site. 53 BP1 seems to be involved in some way in some kind of protective role. So that's at least circumstantial evidence that maybe there's a structure that causes a symmetrical DNA damage in the next S phase. Now, that might be a gap in the original chromosome, or that might be a broken bridge. We don't really know that as yet. The other thing that we'll be doing is trying to biochemically test the model, and it's actually a very difficult thing to do. This is something that Georgie's been doing in the lab. And she's been making a model DNA substrate that mimics a kind of unreplicated piece of DNA. So she started with this molecule here and asked whether the blooms, top 3 and its partners or whatever, can actually, if we go back to this, separate these 2 rings into 2 individual ring structures instead of being connected by this region of DNA that's tangled like this. And indeed, BLM can do that reaction along with top 3. But this time, there's an absolute requirement for the RPA protein. And there's a rather more minor role for RMI1. It stimulates the reaction, but really rather modestly. So in fact, this is a reaction, unlike holiday junction dissolution, that requires RPA, and RMI1 plays a more minor role. And so I'll get back to this idea that maybe the RMI1 protein is a sort of specialized RPA. I don't want to push this too hard, but it may well be that when you have holiday junction-like molecules where you're trying to separate 2 molecules where you branch migrating structures, you use the RPA-like molecule RMI1 and 2. And when you're trying to unwind a DNA structure like a late replication structure, you use the RPA primarily. And we know RPA stimulates DNA unwinding by BLM very substantially. So I'll finish off just by talking about this pitch protein. Sorry, I've been rather too slow. Ask what role pitch might play. It's very brief now. So this was the work primarily done by Seiki Hirano in the lab. And he's been addressing the role of this pitch that sits on the bridges. So pitch is called PLK1 interacting checkpoint helicase, identified by Eric Nigg's lab. It's an SNF2 family protein, a DNA translocase protein, and we've actually shown in vitro with purified pitch that it is a translocase, double-stranded translocase. It localizes to centromeric regions, the region where a lot of bridges are, in metaphase and to these anaphase bridges. And we think it actually binds directly to the BLM protein as well, and that's regulated by mitotic phosphorylation, but I won't go into that. So the question we've asked here is, why does pitch coat the entire length of the DNA? It's always been a mystery. There's no sort of pitch on the DNA here. And however long the bridge gets, pitch is on the DNA all the way along, as is BLM. Now, you might imagine if this was some sort of tangled structure, that you'd have the resolving proteins on the region where you have a problem. But no, whatever happens, the bridge just gets longer and longer, and the protein's coated on there. So one hypothesis that we thought was very naive at the time was that what's different about this DNA, there must be something different about this DNA. Now, I said it doesn't have histones on it. Now, maybe that was the thing that was different. Maybe pitch as a SNF2 family protein actually removes the histones, and maybe that's what its main role is. We don't know. But the other thing that's different about this DNA, unlike these pieces of DNA, is this DNA is being pulled in both directions, you might say. this DNA is potentially under tremendous tension due to being pulled both ways. So we actually started to address the possibility that maybe pitch in some way recognizes DNA distortion caused by the tension on the DNA. So we started to do some single molecule analysis of pitch DNA binding. And this work was really done by physicists in the University of Amsterdam, Erwin Peterman and Gijs Wouter. We made purified pitch, GFP and GFP pitch, having the GFP on both sides, and test the hypothesis that pitch binds to stretched or tense DNA. The DNA is attached to a polystyrene bead via biotin, which is coated with streptavidin, and we use a laser rig, an optical tweezers rig, to actually move the polystyrene beads around and apply tension. So just to show the rig that we use, here's our piece of DNA attached to the polystyrene beads. And what we can do is we can basically move the beads relative to each other and therefore pull the DNA and apply different levels of tension. We flow GFP down the cell, wash it away, and ask whether the GFP protein is sitting on the DNA. And we've asked, does pitch bind stably to the DNA? And if so, is its binding in any way influenced by the force that we apply to the bead? And remarkably, it seems that pitch binding is modulated by the force that's applied to the bead. This is very preliminary, and I have to say, I'm still not sure it's true. But here's a bead which is in a fixed position, and this bead is being moved progressively further away from the other one and stretching the DNA. And as you can see from the fluorescent protein bound to it, as you apply more and more tension to the DNA, pitch binding seems to increase. Now, this could be very artifactual. This could be aggregation of the protein or some other, horrible artifact of it. But actually, it's reversible as well. So if you start with a stretched piece of DNA and release the tension, the pitch protein seems to fall off again. So it's very unlikely to be some sort of aggregated garbage on the DNA. But nevertheless, we still have a lot of experiments to do to prove this. So this is our sort of working model now. Cells go into anaphase. They have entangled DNA structures either at fragile sites or centromeres. Pitch is recruited to the DNA in some way by sensing DNA tension. That's perhaps overstating things a little. And then pitch somehow mediates recruitment of the Bloom's topo3-RMI1 complex, an RPA, which acts to resolve the entangled DNA in some way. Now, we're far from proving this, but this is our sort of working model for what's happening in anaphase and how these structures are removed. So I'll finish by just concluding. We think BLM suppresses sister chromatid exchanges, perhaps by a combination of disrupting early homologous recombination intermediates and then disentangling ones that have formed by holiday junction dissolution. We know that the Bloom's top3-RMI1 complex and this protein pitch decorate ultrafine bridges that originate from centromeres and fragile sites. BLM top3 RPA can decatenate late replication intermediates that could be the source of these anaphase bridges. Pitch is a double-stranded translocase that's required for BLM to bind to anaphase bridges, and we don't know what its role is in that process. It might be nucleosome removal, but it might be direct recruitment of BLM. And pitch might be a molecular tension sensor, but take that with a pinch of salt. So just to thank the people who've done the work. These are the guys that have been in Oxford, and the guys in red have moved with me to Copenhagen, which is nice, and we've got some new people in Copenhagen, and we're looking for other people. So thank you very much for your attention. Yeah, there is. We always seem to see that, and we don't know whether that relates to differential tension across the DNA or what, but we do see that. It might be that the protein is cooperatively being recruited in some way to regions of it, but it's a common observation that we see that it's slightly higher intensity fluorescence near the bead than in the center.
So when you put it under tension, does it change from A to B to Z? What's the transformation?
Well, I mean, this isn't what we've been doing. I mean, this is what the guys in Amsterdam are trying to address, what's happening to the actual structure of the DNA and what might be the key. If you can find the sort of crucial level of tension that recruits the pitch, what is the DNA structure under those conditions? Because that might then indicate what it is that's the recognition feature. And they've been playing around with that a little bit, but as far as I know, they haven't actually confirmed exactly what the DNA structure is at the sort of levels of force that pitch starts to associate with the DNA, but they are interested in trying to do that because we don't assume that it's a molecular tension sensor in that sort of way. We assume it recognizes the transition of a distortion of the DNA in some way, and we would obviously like to know what that distortion is and what that change is, but we don't know that at the moment, and pretty certain they don't.
So in the cytology of the anaphase bridges where you visualize pitch, is this live cell imaging with pitch GFP?
We can do that. We have done that. The pictures I showed you are just regular immunofluorescence for the, which one? Just the regular cytology.
I mean the decoration, because
no, that's not live cell imaging. No.
Oh, okay.
But we have got pitch GFP and we've got BLM GFP and we have looked in live cells. You can see them. Eric Nigg's done a lot of live cell imaging with pitch as well.
Now, the idea I had is, but it's excluded by the way you did the experiment, is if it's live cell imaging and you image this for and open the aperture for a couple of seconds, right?
You're not going to see
These
it.
can whiz around because they have very fast motors. They do Red 54 measured here in Davis by Steve's lab. Goes 300 base pairs a second. So if the shutter is open 10 seconds, you can have oscillations that look like covering the whole molecule, but it's really just looking at the molecule in different locations in that sum of time.
But all the other components look the same. So if that were the case, then all the other guys have got to be piggybacking on this thing moving.
Well, that's no problem. You just
So it's got this baggage with it.
yeah, it's like a truck, one motor and lots of...
It's also relatively homogeneous, so if it is translocation that you're seeing, it has to be tracking, equally along. I think the simplest explanation is that it coats the DNA, but there are alternative possibilities.
So we and others have off the track that when we pull on a molecule of DNA like this, the molecule will begin to unravel at the ends and will generate single-stranded DNA.
Yeah, this is the force that's applied is below the level where that happens. That's something that Irvin and Heiss have looked at in some detail, actually. The amount of tension that they need to apply before you start to unravel the ends, and this occurs prior to that occurring.
Well, the second question was going to be whether you evaluated whether RPA is binding to the anaphase bridges in live cells.
RPA is binding to the anaphase bridges, but not interestingly all over the bridge. We see patches of RPA on the bridges. So it's the only protein that we see that doesn't coat the bridges. So if you imagine that that's a marker of single-stranded DNA, then at least some of the bridges have partial single-stranded character. Obviously, it doesn't have to be a marker of single-stranded DNA. Since RPA binds to BLM, potentially it could be sitting on a protein rather than on the DNA. But the simplest explanation probably is that there are regions of single-stranded character. And the reason that we think that's the case is when you do see RPA in these little patches, you don't see pitch in that region. So there are occasions when there's a little region where pitch isn't bound, and pitch is double-stranded DNA specific. So we think pitch defines double-stranded DNA and RPA defines single-stranded DNA. But again, the evidence is circumstantial.
Do you estimate how much of the bridge is coated with RPA? 10%?
Many of the bridges have no RPA on them at all. Many of the bridges have no RPA patches. But from time to time, 25% of the bridge would be RPA bound. So there might be very extensive regions of single-stranded DNA in some cases. I haven't done the calculation, but it wouldn't be particularly difficult to do the calculation. But I mean, they can be several microns in length, these things. So you're talking about very long regions of DNA. So one assumes you must be unraveling sort of kilobases of DNA from the sister chromatids to form this bridge-like structure. They're not. It can't be a small piece of DNA. Not when you've got in late anaphase, anyway.
To get back to the aging part, are hypomorphs to these genes in the system in mice? Or do they have aging phenotypes? That's the first question. And the other one is, if you have cross-linkers, wouldn't you expect them to make more structures? Like you're talking about your system resolves, and are they sensitive to cross-linkers?
RECQ helicase mutants tend to be sensitive to cross-linking agents, but unfortunately cross-linking agents primarily repaired by homologous recombination, so it's a little bit hard to know whether that's a recombinational repair problem or a replication problem. So it's a little bit difficult, but in fact some of the mutants, the strongest sensitivity you see is actually to DNA cross-linking agents, but that's also a hallmark of recombination genes as well. What was your first question?
The other one was if you make
Yeah. You can sort of phenocopy the Werner syndrome phenotype in mice, not by simply knocking out the WRN gene in mice, which actually has almost no phenotype. But if you actually combine that with telomerase deficiency, so lack of telomerase in the mice plus Werner, you then get a premature aging mouse, dramatic premature aging in the mouse. So Werner is acting as a, you might say, aging gene in the mouse, but you've got to get rid of the telomerase. And that might be because in mice, telomeres are very long for a start, so it might be a telomere phenotype. And also somatic cells have got high levels of telomerase as well in the mouse. But otherwise, you don't see dramatic aging phenotypes. There's a Bloom's hypomorphic mouse. You don't see dramatic premature aging with that, but you do see a very strong cancer phenotype.
I'm sorry, I may have misunderstood you, but you said that the fragile sites or hotspots were breaks. Am I right?
Yeah, fragile sites, well, this is actually slightly controversial as well, but historically the argument has always been that fragile sites break in metaphase. So the classic picture of a fragile site is of a metaphase chromosome with one of the sister chromatids sort of leaning over to the side with what looks like a break. And so that's always been considered to be a broken chromosome. There is an alternative interpretation of that actually it's not condensed. And in fact, you just can't see it. So it looks like it's broken, but in fact, it's not condensed, and therefore it looks like a break. So they may not actually break. They are hotspots for chromosomal rearrangements, though.
Because I thought the reason you thought that the bridges are not as a result of homologous recombination intermediates, but rather delayed replication was, I'm not sure, like if there is no strand break, why wouldn't it be?
Yeah, I didn't say why we think that. That's because, for example, if you deplete RAD51, which should suppress recombination, yeah, and the bridges would derive from recombination, you might imagine that their level would decline. But in fact, the level goes up enormously. So RAD51 depletion causes huge numbers of bridges in cells. So we think that bridges derive from a process you might say that homologous recombination normally suppresses. They're not caused by recombination, they're in a process that recombination normally suppresses. And all the other things we've done with replication, perturbation, and whatever, lead us to think it's much more likely that they derive from a replication problem than strictly a recombination problem. But some of them could be recombination intermediates. I'd love some of them to be recombination intermediates. Don't get me wrong. You know? That's why we were doing it. You know.
So back to the bridges. I mean, are they simply a single duplex, or are they catenated in any way? And then the related question is, when you saw the 3BP1 foci, do those also coincide with H2AX foci at those locations?
Yeah, I mean, to answer that one, they do. You get eight gamma H2AX foci frequently at fragile sites, But again, because gamma H2AX probably isn't highly specific for double-strand breaks, it's a little bit hard to interpret that. But let's assume that that indicates a break. Many times, if you do immunofish on metaphase chromosomes, you get beautiful gamma H2AX foci, rather like the FANCD2 foci, and they're frequently at fragile sites. They're also frequently at telomeres, interestingly, but they're frequently at fragile sites. And so you see those foci sometimes in the next cell cycle. 53BP1 just happens to work really effectively. And the other thing I forgot to say is that there are always these enormously big, juicy guys. They're not little, tiny gamma H2AX foci. They're really big foci. And Yuri, at least, is trying to address why that might be the case. But we think that we don't actually think that they derive from broken bridges. We actually think that they derive from the inheritance of the fragile site problem in the chromosome, not the bridge per se.
And is a bridge just a single duplex? Is it that simple?
Is the bridge a single duplex?
Or is it two? Are they linked in some way?
How do you measure that, you know?
I don't know.
Yeah, if you could come up with a good idea as to how you measure that, I'd love to hear it, but I don't know. I mean, the simplest interpretation from the model is that they would be, essentially a single duplex, and then you unravel the DNA from that. So you might have two duplexes at some stage, but that might be why you can't see them with DNA dyes. It might be just a boring thing that to do with sensitivity of the dye. It might not be that the dye doesn't stain them. It might be that you just can't see it. Immunofluorescence has an amplification event that allows you to see the proteins on there, but of course the dyes, you may not be able to see them. So we really don't know what their structure is.
You said these bridges were pretty common. Should we infer from that that a pair of cells that has a bridge between them, that both daughters are going to live and we have normal genomes after whatever happens?
We think so. I mean, this is the stuff, again, that comes back to what Yuri's been really looking at in live cells. There's no evidence that at least immediately after mitosis something catastrophic occurs and one of the cells apoptoses or whatever. The cells can be followed through the cell cycle. These foci disappear in the next S phase. So we think this is a cleaning up mechanism. We just think mitosis is far less, you might say, efficient than we thought it was before. There are far more problems that are being cleaned up in mitosis. And then very occasionally, you even then go into the next cell cycle and you've still got some garbage left behind that you have to clean up. Then you go into the next S phase and you come around again. So there's this almost constant cleaning up process taking place. And so these things aren't that efficient. Now, obviously, we're using cancer cells, which are abnormal, you might say. But even if you look in normal cells, you can see these bridge structures. And I'd say in early anaphase, they're not actually that hard to find. They're much smaller, and therefore you might say they're a little bit harder to visualize. But they're not that difficult once you get used to looking at them. These late anaphase cells with enormous bridges are rather rare, it has to be said. But you can induce their numbers. There are many, many ways in which you can induce their numbers. So you can end up with virtually every anaphase having many, many bridges. I mean, a good way of doing it is to prevent conventional decatenation process by topoisomerase 2 by using inhibitors of topo 2, ICRF193. Then you get hundreds of bridges, absolutely hundreds of bridges. Almost looks like you've got just a smear of red fluorescence between it, but there's so many bridges that you can have a cell going through anaphase with literally hundreds of these bridges.
We quite have to limit. It's about one for one.
So is there any role of the structure selected in the nucleases like MAS81, SLIX14, YENT1 in processing anaphase bridges?
We're obviously analysing that in some detail. We're particularly interested in yen and gen, of course, because gen 1 is known to be very strongly regulated in the cell cycle, and the transition of the change in its phosphorylation status, dramatic change in its phosphorylation status, occurs in mitosis. So one possibility of the role of yen and gen is actually not as an S-phase resolvase, but as a mitotic cleaning up system. That might be its backup process. That's complete speculation, I hasten to add, but that's a possibility. But we're obviously very interested to find out if these guys have a role.
So just to clarify for myself, and I know you don't know the structural details, but what you seem to have in the back of your mind is that there's single strands wound around each other that are holding these sisters together. It hasn't gotten replicated. You just need a topo 1, type 1 topo to come in and nick it and fix the problem.
No, you need to unwind and decatenate the strands. It was originally shown by Ken Marion's group using coli-RecQ and top3 that you can do that over quite extensive distances, actually. And we're doing it with BLM. It's a very inefficient reaction in our hands in that system. But, Ken had a really very good, efficient system working with Q and topo3. So actually, you can get that to happen. Now, you then got to say that's a dangerous thing to have. It could go around basically separating all of your DNA strands. So that obviously has to be in some ways regulated. But there's no doubt that at least in vitro that reaction can occur. And you can separate really quite large structures of unreplicated DNA. In our case, we've only got we've tried molecules with something like 50 base pairs or things like that. But Ken, I think, looked at structures that were hundreds of base pairs in some of the molecules. And you can get them to be separated. So a helicase plus a type 1 topo can basically just go along a piece of duplex DNA and pull it apart.